Hello dear colleagues, today I want to present you a work on the topic Neuroevolution methods for the organizing the search of anomalies in time series. So let's start. In this slide you can see the compression of the model difference based on human participation and the complexity of the models themselves. It is important to recognize the main idea of the work, and we all know that when we talk about some model which based on the principles of artificial intelligence, the complexity of these models can be more and more difficult. In the next slide you can see the example of time series. We all know that this is some format of input data, and what is more interesting, this data usually can't have the huge amount of some parameters independent, which can in some way change the dependent parameter. And in the slide you can see the very simple examples of time series, and what is more interesting, this time series demonstrate the data from some type of analysis of human body. We all know about it, and today the sensor system can bring the data from the human body 24 hours for the 7 days, every week, every day, and the amount of this data will be so huge and so we need some new approaches that can help us to detect the anomalies in such time series. In this slide you can see the compression of anomaly recognition methods, and you can see the different methods, and all the different methods use different strategies for it. In this case, yes, classification may be one of the most interesting methods and approach, because it can not only recognize some anomalies, but can classify these anomalies. But we all know that classification methods usually based on the supervised or partially supervised learning, and it can be some frames to these methods. So maybe interesting is idea which can help us to walk through these frames and can change some ideas of these methods. In the next slide you can see the main idea of proposed method in the paper. So the main idea is simple, and first of all is a set of models, and the second is a set of data individuals. So each model is a sequence of encoder and decoder levels that describes the input multidimensional signal. The system, or the framework, we can name this, allows to create an ensemble model using an approach similar to the method based on packaging in packages. A whole ensemble model is a set of submodels that work with subgroups of input signals, ensemble models from a set. The work of the solution begins with generating initial group of input signals using correlation. The system then updates the model and groups using the genetic algorithm. In parallel, genetic operators optimized individuals models in each subgroup by making changes to the topology of neural network, or to the maybe model lengths or layer parameters and so on. The end result of this action is an ensemble model optimized for anomaly detection. The ensemble models is defined as flows. A number of papers note that despite different models, it is noticeable that almost all models show a similar set of anomalies, despite changing in the hyperparameters. Of course, the results differ depending on on hyperparameters, but none of the changes significantly affect detection. Because of this, an ensemble model is proposed based on simultaneous division of the search space into subgroups and the evaluation of model for such subgroups. As a result, the models were able to identify more specific dependencies and relationships between the signals. In the next slide, you can see the datasets which we used for analyzing of result of working of our methods. Though this is different datasets, and what is more interesting, the number of anomalies in this dataset are different too. When we talk about the Neuroevolution methods, we usually talk about some metaparameters for this synthesis, and the example of these metaparameters for our work you can see in the slide. The experimental result demonstrates that parallel clustering of data and synthesis of a model based on the proposed data using an ensemble system can significantly increase the efficiency of the anomaly detection process. The process of Neuroevolution helps to synthesize model and develop them into stage based on update information about the input data without separating the process of data preprocessing. Test based on different data sets, and in all cases the result of anomaly detection is satisfactorily raising the quality of level of detection. The improvement of some datasets more significantly than in other datasets because there are more sensors and samples in this dataset. So the amount of dataset is important too. In this work, we provide the Neuroevolution approach that can have a positive impact of the results. In the future, can be more focused on the combination of different solution topologies and using it during processing. At the conclusion, we must say that this work consists two stages: the separation of search space and the synthesis of model. Such approach can give more better results, and in the training stage the method process and separate data about the behavior of the system. In the synthesis mode, the methods gradually adjust the models in order to get the final solution in the future. The resulting model is synthesized using uniform crossover, which makes it possible to increase the size of the parent pool from 2 individuals to a much larger number. This is interesting in this idea because when we talk about the clustering of input data, we can talk about the different number of this cluster and so different number of the submodels which can use this information. And in the future, it can help to organize this crossover based on different models from the different subpopulations. In this stage, this is all, and we thank you for your attention.